Welcome to STAAR Mix 5. We're going to cover the cell cycle in its entirety, including mitosis and cancer. At this moment, the cells in your body are growing, dividing, and dying. All the cells in your scalp, palms of your hands, soles of your feet, they're all shedding and being replaced. As cuts and bruises are caused, so they have to heal. And your intestines are actually producing millions of new cells each second. But what would happen if your body couldn't make new cells? What would happen if your new cells did not have all the right amount of DNA or organelles? What would happen if your cells kept duplicating over and over and over again without end? Those are the questions that we're going to talk about in this first half of the office mix. Based on this image, what should happen as the result of the cell cycle? What do you notice about the cells? You have the parent cell here, and you have two daughter cells down here. Are they identical, or are they different to the parent cell? If you scratched your cheek, would you want the cells replacing the dead cells to be identical to what they were before? Do you want your cheek to grow back and heal the same way? This is the whole idea behind the cell cycle, replacing old and dying cells with identical new fresh cells. We're going to do a quick review of DNA. Your DNA is found in the nucleus and it never leaves. Your DNA is the code that makes up who you are. It's unique to you. It's made up of a series of nitrogenous bases, the A's, T's, C's, and G's, as seen here. The order or pattern that they make is unique to you. DNA is found in two forms inside the nucleus. It's found in chromatin form. It will either be unwound and stringy in this form, chromatin, or tightly wound and packaged in a form called the chromosome. The chromatin is wound around protein structures called histones, and you can see the histone here. The histones fold and pack together over and over and over again until the chromosome structure is formed. Here's a look at the noodle soup that is chromatin. When it's unwound, your DNA is massive. During G1, this is the form that your DNA takes. Here's a look at the shape of a chromosome. This carries all of your genetic information, and during the lecture we're going to talk about and identify what form the DNA will be in at each phase of the cell cycle. The cell cycle is broken into three separate phases, and each one has its own specific purpose. So we have one entire cycle called the interphase. This is part 1. Part 2 of the cell cycle is mitosis. This is where nuclear division happens. And then the third part is cytokinesis, and this is the division of the cytoplasm of the cell. The majority of a cell's life cycle is spent in the growth period known as interphase. Interphase is the busiest and longest phase of the cell cycle because this is when the cell actually prepares for division, which is also known as mitosis. Interphase itself is actually divided into 3 stages, G1, S, and G2. The arrows here are kind of hard to see on the circle, but the cycle here is going clockwise. So here's an arrow, and the cycle is actually going in this direction. At this point, the next cycle, mitosis, starts, and at this point, cytokinesis starts. G1 is the first growth period. You can kind of think G equals growth, first time, so growth for the first time. All normal functions happen here. But what are normal functions for a cell? Well, creating energy, cellular respiration, creating proteins or protein synthesis. These are both normal cell functions. The cell is also going to synthesize extra proteins called histones. Remember those? Those are needed for when new DNA is created later. G1 phase is really important for the cell because it also decides here if it wants to move on and create a new cell or if it wants to remain dormant, i.e. in G0, which is the dormant phase of the cell. During G1, DNA is in the long stringy form called chromatin. It needs to be in this form so that transcription, or the creation of mRNA, can occur in the nucleus. If it was wound up into the chromosome form, the individual genes could not be transcribed into the mRNA. The next phase is S. This is when the DNA is synthesized or replicated. S equals synthesis. Anytime you synthesize something, you are creating it. Here we have to create an exact copy of the original DNA strand in the cell. So for a while there will be exactly 2 copies of the DNA in the cell. This information is used in STAAR questions all the time. Just remember that if you're starting in G1, you have 1 times the material, the original material, and in the S phase you double the amount of material. This doubling of the material will remain until the mitotic stage, when it actually gets separated into 2 individual cells. The DNA is still going to be in the stringy form called chromatin. DNA replication has to occur before the cell can divide in 2, and remember the stringy form is the easiest form in order for this to happen. You're going to start with 1 DNA string, and this is going to duplicate with a centromere holding the 2 together. As we go through the next couple of images, I want you to label the image on your paper. The 2 halves of the structure are called the sister chromatids, as they are identical. Label them on your paper. The sister chromatids are held together at the center by the centromere. Go ahead and label that on your paper. Again, why is it important that the new copies of DNA are exactly like the original? What are we trying to accomplish eventually? We're trying to make new identical daughter cells, but in order for that to happen, every new cell has to have an exact copy of the original DNA. Now we're moving on to the final step of interphase, and that is G2, also known as the second growth cycle. Here, the cell organelles are going to be duplicated. So a good way to remember this is growth equals G, or G is the first letter in growth, and 2 equals double organelles. So again, the cell is going to grow bigger, but we're also going to be duplicating all of the cell's organelles. So now that we have finished interphase, we have a cell that is twice the normal size, has duplicate DNA, duplicate organelles, and plenty of room. What was the point of interphase? What are we doing here? Well, we're preparing to divide the nucleus and eventually the 1 cell into 2 cells. The next step in the process is mitosis. So if we are creating a whole new cell, why do we need an exact copy of DNA? Why can't we just take a little bit of DNA from the original cell and, like kind of spread it out like peanut butter and jelly? Because every cell must have the exact same code and therefore the exact same amount of DNA as the parent cell. This exact code has to be letter by letter identical, or else the cells will not create what is needed, the proteins needed, to continue the life processes of the organism. Mitosis follows directly after the completion of G2. The purpose of mitosis is to divide the nucleus. It's important to associate mitosis only with nuclear division. Mitosis is divided into 4 phases, and an easy acronym for the phases is PMAT, P-M-A-T. Prophase, metaphase, anaphase, and telophase. Follow along in your worksheet as we go through each of the phases. During prophase, the membrane around the nucleus is going to begin to dissolve. Centrioles are going to begin to move to the opposite ends of the cell, and the spindle fibers begin to form. This is considered early prophase. You can still see the nuclear envelope here, but during prophase, this is going to disappear. During late prophase, the chromatin is going to wind up tightly into the fully replicated chromosome. Why do you think this needs to happen? I want you to remember that the chromosome is about to be split apart, and although it's hard to imagine based on this picture, the chromatin strands are actually really, really long. Imagine for a moment that you work at Hobby Lobby and some person came into your store overnight and unwound all of the spools of thread. Your boss comes to you and says, "I need you to divide all of this string exactly in half." Would you be able to do it? Or would you look at him like he's crazy? Now imagine that all the string was tightly wound up around spools of thread, like it normally is, and he says to you, "We need to divide all the spools exactly in half." Would this be more manageable? This is the reason that chromatin winds around the histones, or the spools, into the tightly packaged chromosomes. It's so that when it pulls apart, it's a manageable separation of the DNA. This is the later part of prophase. The nucleus is completely dissolved and the chromatin has packaged into the chromosomes. Metaphase is easy to remember because the chromosomes line up in the middle. You can also say that they meet in the middle for extra emphasis. This is also one of the easiest phases to recognize. Once the chromosomes line up, the centrioles send out spindle fibers that will attach to the centrioles, or the center structure of the chromosome. Here are the centrioles, and you have your spindle fibers here, reaching out to attach to the centromeres. Here is the middle, or the equator of the cell, where the chromosomes are lining up. Notice here is the individual chromosomes. You can get an idea of the centromere at the center. These two lines here represent the spindle fibers and how they attach. This chromosome is going to be pulled apart in this direction. And the reason this is so important is because you need these duplicate sister chromosomes to have one in one cell and one in another, so that the DNA is identical in each cell. Here's a look at metaphase under the microscope with clearly visible spindle fibers. Here's the chromosomes meeting in the middle, and here are your individual spindle fibers. You can almost kind of make out the centriole right here, these little black dots. During anaphase, the spindle fibers pull on the centromeres, breaking them, and pull the sister chromatids apart to either side of the cell. The sister chromatids are pulled towards the centrioles. This phase is remembered as A for apart, because the chromosomes are pulled apart. Keep in mind that the way the chromosomes are divided makes sure that each cell gets the same identical genetic information. Here is anaphase. It's recognizable by the V shape of the chromosomes as they're being pulled. The final phase of mitosis is telophase. During telophase, you begin to see the formation of two cells. They're not divided yet, though. The chromosomes unravel back into the chromatin as they're no longer needed to be packaged because everything is already separated. Then the nuclear membrane begins to reappear around the chromatin. Keep in mind T is the first letter in two, but they're not actually separated into fully two yet. You just can see the outline of two individuals beginning to form. Here you can see the beginning of the nuclear membrane. This completes the 4 steps of mitosis: PMAT, prophase, metaphase, anaphase, telophase. The final phase of the cell cycle is cytokinesis. It starts directly after telophase. Try to think of cyto as the prefix of cytoplasm. We will be dividing the cytoplasm completely, and at the end of this stage we will have 2 separate cells. Cytokinesis differs between plant and animals. In animal cells, the plasma membrane is going to begin to pinch and look almost like a peach. It'll begin along one side of the equator and continue until the entire cytoplasm is split in two and there are 2 completely separate cells. Here's the step-by-step. In this image, it actually begins in anaphase, and then telophase, then cytokinesis in 2 steps. But I like the close-up of what is called the cleavage furrow because it looks like a messed up peach to me. And this is the cleavage furrow here. So in plant cells, we have a unique structure, a cell wall. Plant cells have a rigid cell wall, so the plasma membrane isn't going to pinch. Before cytokinesis can begin, the cell plate must be laid down across the equator. Cytokinesis is complete whenever the 2 new primary cell walls have formed, and it's clearly 2 separate cells. They're going to stay side by side, though. Single-celled organisms like bacteria can go through mitosis, and it's typically called asexual reproduction or binary fission. And when they go through mitosis, they're going to remain as single cells. In multicellular organisms, mitosis happens in the body cells, also known as somatic cells. It happens all the time. This is how a child grows, and this is how you make new cells to replace the old ones or ones that have been damaged. These cells work together to form tissues that have specific function in your body. Each new cell will be identical in every way to the one that came from, and will be immediately ready to work along with the tissues and organ systems that it was specialized for. However, occasionally cells lose control of the cell cycle. Cancer is the growth resulting from uncontrolled cell division. Cancer can be caused by environmental factors such as carcinogens like cigarettes, too much alcohol, even your lifestyle. Your body is a delicate machine, and whatever you do to it or put in it could potentially harm the cells. There are also some viruses that can cause cancer, such as the human papillomavirus or HPV, also known as genital warts. It can damage the cell cycle of cervical cell tissues. There are also types of cancer that you're genetically predispositioned to, and these can be tested for. For example, you may have a family history of colon cancer. This doesn't mean that you're going to get colon cancer, but it may mean that you have or carry the gene for colon cancer. It may be that even some genetic cancers are triggered by environmental factors. There are enzymes, which are proteins, that control the rate of the cell's life cycle. And if something goes wrong with these enzymes, the cells will skip a long interphase and divide a lot faster than they should. Basically, it'll be a continuous mitosis cycle. So you can kind of imagine if it went along the growth and replication phase and kept going back through mitosis over and over and over again. The cell cycle has to pass through certain checkpoints to continue on through the cell cycle. If the cell fails a checkpoint, this usually means it'll be damaged or growing incorrectly. It will either be repaired or it will perform apoptosis or cell suicide. The G1 checkpoint is very critical. At this point is when the cell determines if it is going to divide at all or if it will go into G0, which is the non-division stage. G2 is also a critical point, where the cell checks that all the DNA has been correctly synthesized and that the organelles are duplicated and the cell is big enough for division. If either of these checkpoints are skipped, the cell is going to be stuck in a never-ending mitotic cycle, creating never-ending supply of cells. Cancer cells are cells that have been damaged and escape these checkpoints. Because they're no longer regulated, they don't go through the cell cycle at a normal rate. They have the ability to divide infinitely. Most normal cells have a maximum amount of times they can go through the cycle. Cancer can be benign, which means it doesn't spread through the entire body. As the cells divide, though, they need valuable nutrients, just like any other cell does. This mass that you see here is referred to as a tumor. This tumor creates a mass of cancer cells that are going to constantly divide and form this mass, but they're also going to suck nutrients from the healthy cells and the healthy tissues within the organ system. So yes, cancer can be benign, which can be removed, typically depending on where it is in the body, but cancer can also turn malignant. This means one of the cells or more have traveled to other parts of the body through the bloodstream. Once this happens, it's much more difficult to treat. There's a couple of different current cancer treatments. There's treatments that are chemical that target rapidly dividing cells. Unfortunately, this will also target your hair follicles, which are also rapid dividing cells. You can also use radiation if it's going to target a large tumor or buildup. There's also medications that are being researched that may cut off the blood supply to tumors, which is really important because part of the reason why cancer is so harmful is because it sucks nutrients from other parts of the body and organ systems.